Hey everyone, we are in the chapter 1 notes, slide 13. We are going to talk about the different ways to study anatomy. So what are the different ways to study anatomy? Well, there's several ways. Let's start with an example. Here we have a brain. I hope we can all agree with that. This is a brain. What are some words that we can use to describe this brain? Some objective observations about this. We can say that it is wrinkly. We can say that it looks a bit soft, maybe a little slimy or wet. You might have some other terms to describe this brain, but I think those three are good to start with. In this class, of course, we're going to study how the structure of something relates to its function. That applies to anything from this pen to body structures. So a pen is long so that it can fit in my hand, and small and round so it can fit in my hand. Why is a brain shaped the way it is? Well, let's review, what is a brain's function? A brain's function is to process things. So let me write down here, its structure on one side, its function is to process, to control, to communicate. We can come up with more words of things that the brain can do. Whoops. But this list will do for now. So how does the brain being wrinkly, soft, and slimy relate to it being able to process, control, communicate? The fact that it's slimy, it's surrounded by liquidy stuff. We're not going to get into that right quite yet. We will a bit later. I think the thing that will stick out the most right now is the fact that it's wrinkly. Why is the brain so wrinkly? By being really wrinkly, you can fit more brain matter into a smaller space, into a smaller volume. Maybe you've heard the term increasing surface area relative to volume. We're increasing the surface area to volume ratio. All that is saying is that we're fitting more stuff efficiently in a smaller space. If we want our brain to be able to process and control and do a lot, we want to be efficient about it. If our brain were not so wrinkled and we wanted the same processing power, our brain would have to be huge. And imagine having to carry around a really huge brain. That would be very, very inconvenient at the very minimum. It would take a lot of energy to do, and we already use a ton of energy. So we want to have small heads, relatively small brains, yet pack a lot of efficiency into it. So the brain is wrinkly so that we can fit more and do a lot with the size of brain that we have. Now we can do the same thing with what we see here. This, if you haven't guessed it, is a neuron. There's actually two here. But you can see here's one part of the neuron and it branches out. So let's do the same thing we did last time. What are some structural terms we can use? I can spell, I can do it. There we go. What are some terms to describe the structure of a neuron? It branches a lot. I would say that's the main structural term we can use to describe the shape of a neuron. A neuron branches a lot. In terms of its function, communication. How does the structure of a neuron relate to its function? By having branches, branches allow neurons to receive input and send output from multiple or to multiple places. with other neurons, with other targets. We'll get to all that later. We'll just put with other cells for now. Other places, other cells. So what we've seen here is that whether we're looking at the brain, which is a gross anatomy, large structure, or the neuron, which is microanatomy, a very small structure, relatively, regardless of what level of organization we're looking at, we can always study how the structure of something, the anatomy of something, relates to its function. And that's what anatomy is. Anatomy is the structure, the study of morphology, and studying how something looks and relating that to its function. That's what physiology is. Physiology is the study of the function of the body. How does it work? There's always a relationship between structures and functions for biological systems like ours and every other living organism, and for inanimate objects like this pen. We, of course, will be focusing mostly on anatomy. We'll be studying basic functions of things, just like why is the brain shaped the way it is? Why is the neuron shaped the way it is? You'll be going into way more depth in physiology when you take physiology about how exactly things work. We go into really detailed mechanisms. So we've seen already that we can study anatomy in terms of gross anatomy and also microanatomy. So let's look more closely at how can we differentiate different ways to study anatomy. Using our eyes, using gross anatomy, we can study different body parts. We can call that regional anatomy. For example, let's say you are a doctor that specializes in the hand. So you know all about, you know about everything else, but you really know all about the hand. The hand has a lot of different things. You have muscle, you have blood vessels, you have bones, you have nerves, and many other tissues. It's very intricate. We could study specifically just about those things in one region. That's something that typically is done in medical school. We're not going to take a regional approach, but sometimes we do, just a heads up. That's one way you can study anatomy, using a regional approach. What we will focus on mostly is a systemic approach. A system, the word system here is referring to the different organ systems. You may know these different organ systems like the reproductive system, urinary, digestive, respiratory, cardiovascular, endocrine, nervous, muscular, skeletal, and integumentary. If you're curious, integumentary means skin, hair, nails, and glands that go in your skin. That's what systemic anatomy is. We will be focusing largely on systemic anatomy. Initially, we need to study the basics of different terms of anatomy. We'll be studying cells. And once we get past cells and then tissues, we can get to our systems, starting with the integumentary and going on from there. So that's another way that we can study anatomy based on systems. We call that systemic anatomy. I want to emphasize the importance here of all these systems, that while we divide our systems, let me go back, while we divide our systems into different parts, cardiovascular, respiratory, digestive, whatever, these systems are all interrelated. Sometimes they overlap. For example, you think of the heart as cardiovascular, but the heart is actually also an endocrine organ. It does produce hormones. That's something we'll get into later, maybe not at all, maybe something you'll study in physio. But there is overlap, and all these are interrelated. They communicate with each other. This is a really simplistic view of the human body. Like, imagine this being your skin, all this outer stuff. Imagine this is where you breathe. So this is where your lungs are. When you breathe, for instance, you breathe in oxygen. Oxygen gets into your bloodstream, and then oxygen gets to your cells. When you eat food, food goes in your mouth, stuff gets into your cardiovascular system. Some of that will get transported to your cells, but then some things will get excreted out of your urinary system. Some things just continue out through your entire digestive system out the anus. They all communicate with each other. All these systems communicate and exchange with one another in some way. Another way that we can study anatomy is based on surface features, landmarks, surface anatomy. So this is an example of how we can study gross anatomy. Say you look at someone's finger. When you study someone's finger, you can see different joints. You can see different regions of the nail. We'll get to all these different terms later. Don't worry about that right now. We can name different parts of the body, which we'll be getting into in the next video or the next one after that. We can study surface features. It's useful for looking at different landmarks, of course. If you want to identify, say I want to tell my doctor that I found a mole somewhere, somewhere on my arm. Well, my arm is really big. Which region of my arm did I mean? Did I mean the forearm, the antebrachial region, my elbow, the olecrational or antecubital region, the brachial region, deltoid, axillary? What part are we talking about? Surface anatomy allows us to identify things on our surface, skin, hair, nails, and any other things that might be going on. If you want to study an individual cell, that's what cytology is. Cyto means cell. Ology means study of. Oops. Cytology, the study of cells. So we could study individual smooth muscle cells or oocytes. That's just a fancy word for an egg cell. Sperm cells, neurons, adipocytes. Another word for adipocyte is a fat cell. Cells that store fat. You could study individual cells. Since we study anatomy, we rarely find cells separated from one another. The term for cells that are instead grouped together, that's a tissue. A tissue is a collection of cells of the same kind. When we study tissues, the term for the study of tissues is histology. So histo is referring to tissues. This is also like before with cytology, you need a microscope for this. To see epithelial tissue, that's one tissue type, nervous tissue, things like your brain, spinal cord, nerves. Connective tissue, things like blood or bone or fat. Muscle tissue, like stuff that moves your bones or your heart or in your digestive tract, for instance. These are all examples of different tissue types, and to study them at a microscopic level, that's what histology is. We're looking at histology of different tissues in these four pictures. What I want to point out is that they all look different. Well, let me back up here. I know that maybe you're not used to seeing pictures like this and they all look the same. They're all this pinkish hue. It will take time for you to get used to seeing histology pictures and deciphering these small details. Because since I'm practiced, I can easily identify that the bottom right is muscle, the top left is epithelial. I'll teach you specific clues for you to help you figure out what you're looking at. So if these do look the same to you, don't worry, we will get there. But we're going to look for those slight differences in these pictures for you to figure out which one is which and why they look the way they do. For instance, we already saw why nervous tissue is nervous tissue. By having these branches, it can communicate with other cells. Something that we'll be looking at when we look at histology is, well, the thing is, when you look at histology, we're looking at slices. We're looking at things in 2 dimensions. In order for you to see a tissue, you have to slice it and put it on a slide, something we'll practice or look at later. If you slice a banana, for instance, something else we'll do too, if you slice a banana length or so that you cut it like that, that's one way to cut a banana. You can just see a circular part, but you can slice it the long way. You can also slice a banana like this. Those are 2 examples of how to slice a banana. If you slice it the way that you get the round part, we call that a cross section. Cross sections are cuts that are perpendicular to the main plane. A longitudinal section is a cut parallel to the longest dimension. They're two different cuts of the same thing, and they look completely different. What we see here, these two pictures on the left side, let me use a different color. This is skeletal muscle. And each of these circular things, that's a skeletal muscle fiber, a cell. They look circular. But in this picture down here in the longitudinal cut, they look really long. Because like a banana, these are long structures, they're long cells. This picture on the far right shows how, it's an illustration, shows how muscle fiber, it's cylindrical shaped, is really long. So if you cut it cross section, you get a circle. If you cut it longitudinal, you get these long, fairly rectangular structures. That's if you cut it perfectly at a right angle. But what if you cut it at a diagonal? If you cut it at a diagonal, you'll get oblique fibers. And these cells aren't perfectly arranged all the time. Often these cells, some are going this way, some are going this way. So when you cut it, sometimes you'll see longitudinal fibers, sometimes you'll see cross sections. Sometimes you'll see both in the same section. This will take practice for you to identify what it is you're looking at. The cells you see here, this bottom right picture, they're all the same cell type, but they look different shapes because they're cut in different ways. So it will take time, and we're going to get practice that you need to recognize that you're looking at a two-dimensional thing in a three-dimensional world. Picturing how we can piece together two-dimensional things and make something that has three-dimensional volume. Another way that we can study anatomy is developmental anatomy. Developmental is where we're looking at the progress. Sorry, there we go, slide 24, where we're looking at the progress of development over time. That's the key phrase there, something happening over time. So we have an initial point where we check something, and then we check it later, and then we check it later. We could have at least two time points, preferably multiple. You can see how something changes. In this animation, we see a zygote. We're going to learn this term later. A zygote is a fertilized egg where sperm fuses with the egg. Then it goes through rounds of division, more and more division, until it starts dividing into these weird structures. We're going to learn these different forms of development of an embryo, until eventually you get to a fetus, and then that fetus leads to the birth of an infant. That is one example of developmental anatomy, looking at the whole development. We can look at the development of individual things, like something that we'll be looking at is the development of bones. Bones don't just immediately pop up. They have to grow in. And even after you're born, your bones are still growing. So we'll look at developmental anatomy in specific instances. Since our anatomy is so close to the anatomy of many other organisms, especially mammals, and because mammals are easier to come by, and in terms of getting organs or slides preparations, we often look at the anatomy of other organisms to study our own. The similarities are very, very close. We're going to point out some key differences, of course, but the similarities are there. One of the first things we're going to be doing is doing a fetal pig dissection. Pigs have a very similar body plan to ours. They have the same organs, respiratory organs, the lung, spleen, digestive organs. They have all the same organs that we do in roughly the same plan that we do. They're, of course, four-legged. We're two-legged. Slight differences there, of course. Slight differences make all the difference, though. But when it comes to studying anatomy, it's still very useful. Even at a microscopic level, so both gross anatomy and microscopic anatomy, we can study differences. Looking at human sperm versus mouse sperm, they look almost identical. Maybe slight differences in the size of the head, but they look really, really similar. If I cross this out, even I would have trouble figuring out which one is mouse and which one is human. Down here, this isn't sperm, this is something else. We're looking at epithelial tissue. And you can see these groupings of cells here and here. I know they're different colors, that's just a different staining, but these are both the same cell types, and it's really hard to tell, as at least from me, since I'm not an expert in human versus mouse histology, well, for these examples, it's hard to tell which is which. So it's still very useful to study the anatomy of other organisms in order to study our own. More for a convenience. If we do want to study human anatomy without cutting into someone, one great way to do this is using new special, when I say new, I mean in the past 100 years, new tools to visualize sections of the body. If we don't want to cut into someone's body, we can, with these special imaging techniques with radiographic anatomy, we can look at sections. So just like those slices of tissues you've seen before, we're getting slices of gross anatomy. We can use X-rays, CT scans, MRIs, ultrasounds. All these things allow us to see inside a human body. And even this, like before, you need to visualize how we are looking at something that's two-dimensional and imagine what it's like in 3 dimensions. When you see this picture of the abdomen, it doesn't look like an abdomen, but you can see here's the stomach, here's the liver, here's a rib, here's a vertebra. Identifying these things allow me to get some sense of direction. This is the front, this way is the anterior side, this way is the back, this is the way is the posterior side. Knowing what you're looking at can help you visualize where you are. And of course, we're going to get practice with that. So like I said, we're going to be studying anatomy at multiple levels of organization. When I say levels of organization, what I mean is studying things at a very, very small level, like individual molecules. One example of a molecule that we'll be looking at a lot are different proteins. Proteins are made out of amino acids, but proteins. An example of a protein that maybe you've heard of is hemoglobin. Hemoglobin is found in your blood cells. This protein shown here, actin, this is a protein that's used in your muscles to move them. So we'll be studying individual molecules. Molecules are what make up cells. Cells together make up tissues. So if we have, let's say, proteins inside of a muscle that makes up the heart. The heart is an organ. The heart is part of the cardiovascular system. So that's an organ system. And then when you have all these systems together, that makes up an organism. We're going to build our way up from cellular to organ systems. And regardless of what level we're looking at, like I've said before, whether we're looking at a brain would be an organ level, or a neuron, that would be cellular level. No matter what level of organization we're looking at, it still applies that the structure of something relates to its function. Like I said, the structure of something relates to its function. Neurons are branched, so communicate. Brains are folded so that you can have more brain in the smaller space. It's more efficient, more energy efficient. Yeah, so multiple ways we can study anatomy. Gross anatomy, microscopic anatomy, study histology, surface anatomy, development, using X-rays and other radiographic anatomy to investigate what's going on inside of someone. Next video, we'll be going over more language of anatomy, like I use those terms anterior and posterior. How do you use those terms and other similar terms? We'll be going over that, as well as body regions and cavities and all other kinds of fun stuff. So see you in the next video.